students. Myself Devina, working as assistant professor in Science College of Pharmacy. So today I want to discuss with you about the ultrastructure of bacteria. Before going into the detailed discussion of bacteria, we shall see a quote on bacteria. So tiny bacteria are the architects of life, shaping the ecosystem and driving the cycles of nature. Microorganism and all the living organism which is being present in the world, it is being divided into two categories, prokaryotes and eukaryotes. So bacteria is an unicellular organism which is coming under the prokaryotes. So we should come to know what is prokaryotes are. The size of prokaryotes is 1 to 5 micrometer in size, and it undergoes asexual reproduction, and it does not undergo mitosis and meiosis. There are two types of bacteria which is being present. One is the useful bacteria and next is the pathogenic bacteria. So useful bacteria is nothing but which is being present in our gut, which helps in the maintenance of our gut, and pathogenic bacteria are nothing but which is going to cause the disease in the human beings or animals. So we go into the discussion of ultrastructure of bacteria. So the bacteria consists of an outer layer called as a capsule. So capsule is nothing but it is made up of a glycocalyx. So glycocalyx is nothing but it is a polysaccharide which is a sugar core which is being secreted by the bacteria, and it is being protecting as an outer layer, and this we call as a capsule. So what is the function of the capsule? And a layer for the bacteria, and the bacteria is also having another protection layer called as a cell wall, which is going to give the shape to the cell. It is made up of a peptidoglycan layer. Based on the thickness of the peptidoglycan layer, the bacteria is being divided into gram-positive bacteria and gram-negative bacteria. The peptidoglycan layer is more thick in case of gram-positive, where it is very thin in case of a gram-negative bacteria, and the function of the cell wall is to maintain the rigidity of the cell, and it gives having another layer called as a cytoplasmic membrane. This cytoplasmic membrane, it is being present in both prokaryotic and eukaryotic membranes, and they appear like two layer structures. The phospholipid molecule are arranged in two parallel rows. The functions of cytoplasmic membrane is that it maintains a favorable intercellular osmotic pressure for the cell, and the plasma membrane houses a number of specific active transport systems. It contains enzymes capable of catalyzing the chemical reactions that break down the nutrients and ATP. Bacteria is having a non-homogeneous aqueous medium. The aqueous medium is nothing but it contains 89% of water, and along with water it contains many inclusions. Inclusions are nothing but it is the storage of carbohydrates, proteins and fats. So, and this bacteria, it is having a DNA material. The DNA present in the bacteria, it is being present in the form of a nucleoid. So in case of prokaryotes, we doesn't say it contains nucleus. Prokaryotes, that means bacteria, they do not contain the nucleus, but they have the genetic material in the form of the nucleoid, which is being present in the center of the bacteria. So there is also an extra chromosomal material which contains the genetic material of that particular cytoplasm, and that we call as a plasmid. Okay. Next part we are going to see about the ribosomes. So ribosomes are nothing but which helps in the protein synthesis, and also we are having another structure called as a flagella. So flagella are nothing but they are hollow helical structure which helps in the motility of the bacteria, and another structure we are having is called as a phyla. So the phyla are being present all over the bacteria. So what is the use of phyla? So phyla we call as an sex phylus. So it helps in the DNA transfer from one bacteria to another structure called as a mesosomes. So mesosomes are many bacteria, especially gram-positive bacteria. It possesses a characteristic feature of invaginations, or we call as infoldings, which are present in the form of convoluted tubules and vesicles.